Hi all, welcome to Physio Exam Buddy. This video is on the chemical regulation of the respiration. The chemical factors regulating the respiration are basically partial pressure of carbon dioxide, partial pressure of oxygen as well as the pH of the arterial blood. The chemical regulatory mechanisms, they adjust the ventilation in such a way that the arterial PCO2, it is normally held constant at about 40 millimeters of mercury, and arterial PO2, it is not allowed to fall below 60 millimeters of mercury, and the changes in the pH is opposed. Chemical regulation of respiration, it operates through the chemoreceptors. And in our body, there are two types of chemoreceptors, that is peripheral as well as the central chemoreceptors. So what is a chemoreceptor? A chemoreceptor means it is a type of receptor that responds to change in the chemical composition of the blood or any other fluid around it. Now, we will see in detail about the peripheral chemoreceptors, which is the carotid body as well as the aortic body. The carotid body, it is located at the bifurcation of the common carotid artery into external as well as the internal carotid artery. And it is supplied by a Herring's nerve, which is the branch of the ninth cranial nerve. Whereas the aortic body, it is located near the arch of aorta and the nerve supply is from the 10th cranial nerve. And these peripheral chemoreceptors, they respond to stimulus like hypoxia, that is whenever there occurs decrease in the partial pressure of oxygen, or hypercapnia, that is whenever there is an increase in the partial pressure of carbon dioxide, and in case of acidosis, that is increase in the H plus concentration. So these chemoreceptors, they will sense all these stimuli, particularly hypoxia, which is the main important stimulus, and they send the impulses to our respiratory centers, thereby adjusting the ventilation. Each carotid or aortic body, they contain two types of cells, that is type 1 and type 2 cells enclosed in a capsule, and they are in close contact with the fenestrated sinusoidal capillaries. This type 1 or glomer cells, they are chemosensitive cells, and they have granules which contain the neurotransmitters like dopamine as well as acetylcholine. The type 2 cells are the glial-like and each surrounds 4 to 6 type 1 cells. They function as the sustentacular or the supporting cells. Next is about the mechanism by which these glomer cells are stimulated. So this is a structure of a type 1 glomer cells, and you can see here that there is a potassium channel which is oxygen sensitive in the glomer cell. So the type 1 glomer cells, they contain an oxygen sensitive potassium channel, and they are nearly located near to the arteries. So whenever there occurs a decrease in the partial pressure of oxygen, that is whenever there is hypoxia, these oxygen sensitive potassium channels will be closed, and it causes the decrease in the potassium efflux. So what happens when the potassium efflux is decreased? The charge inside the glomer cells become more positive, that is depolarization occurs. So whenever there occurs depolarization inside the type 1 glomer cells, it will causes the opening of an L type. L type means long lasting opening of the calcium channels. These channels will open, which is also present on the surface of the glomer cell. And opening of this calcium channel will produces calcium influx into the glomer cells. This is the diagram you are supposed to draw along with the chemical regulation of respiration. So the depolarization of the glomerulus cell, it opens up the L-type calcium channels leading to calcium influx, and the calcium influx will trigger the release of the neurotransmitters from the granules which is present inside the glomerulus cell. Early studies have shown that the neurotransmitter released is dopamine as well as acetylcholine. But the most recent studies have shown that the neurotransmitter released from the glomerulus cells is the ATP. And these neurotransmitters then will bind to the receptors and it will excite the nerve endings. And finally, the afferent impulses stimulate the DRG neurons in the medulla, increasing the ventilation. So this is the mechanism of action by which the peripheral chemoreceptor senses hypoxia and how the ventilation is increased. This hypercapnia and acidosis will also activate the glomerulus cells, that is mainly by inhibiting the potassium channel. The mechanisms involved in the inhibition of the potassium channels are either by activation of the heme containing protein or by cyclic AMP formation or generation of reactive oxygen species and decrease in the intracellular pH. So by these four mechanisms, these hypercapnia and acidosis can inhibit the potassium channels and finally getting activate our glomerulus cells. These carotid bodies, they have a very high blood flow of around 2000 ml per 100 gram of the tissue per minute. This is roughly 40 times that of the blood flow to the brain. So, consequently, what happens is that they have a very small arteriovenous oxygen difference despite a very high metabolic rate. So, they mainly respond to arterial rather than venous partial pressure of oxygen. And you should also keep in mind that the peripheral chemoreceptors, it is the only site in our body that detect the changes in the partial pressure of oxygen and hence responsible for all the increase in the ventilation occurring in response to arterial hypoxemia. And the blood flow, because of this enormous blood flow per unit of the tissue, the oxygen needs of the cells, they can be met largely by the dissolved oxygen alone. Therefore, the receptors are not stimulated in conditions such as anemia or carbon monoxide poisoning, which is an example of anemic hypoxia, in which the amount of dissolved oxygen in the blood reaching the receptors is generally normal, even though the combined oxygen in the blood is markedly decreased. So, in case of anemia as well as carbon monoxide poisoning or any condition that is causing anemic hypoxia, the peripheral receptors are or peripheral chemoreceptors are not at all stimulated, which can be asked as an MCQ question. And these peripheral chemoreceptors are stimulated only in hypoxic conditions like hypoxic hypoxia, stagnant hypoxia, as well as histotoxic hypoxia or the cyanide poisoning. And the drugs like nicotine as well as lobelin is found to activate the chemoreceptors, that is the peripheral chemoreceptors. This elevated PCO2, it stimulates the peripheral chemoreceptors, but the major effect of this elevated PCO2 is on the central chemoreceptors. So please keep in mind that the peripheral chemoreceptors are the only site that detect the changes in the partial pressure of the oxygen, whereas elevated PCO2 has its major effect on the central chemoreceptors. Next, we will study on the central chemoreceptors or medullary chemoreceptors. They are located bilaterally on the ventral surface of the medulla, and recent evidence suggests that additional chemoreceptors are located in the locus coeruleus, solitary tract nuclei, as well as in the hypothalamus. Although these chemoreceptors are located in the medulla, they are separate from the DRG and BRG. This DRG and BRG, they are mostly related to the neural regulation of respiration. The central chemoreceptors, they are actually sensitive to the H plus concentration of the CSF as well as the brain interstitial fluid. So this is the only direct stimulus inside the brain which stimulate the central chemoreceptors. But the problem is that H plus cannot easily cross the blood-brain barrier. Then how come it will stimulate or detect the PCO2 changes, and how the central chemoreceptors are stimulated? So it is the carbon dioxide that penetrates the blood-brain barrier, and the carbon dioxide forms an indirect stimulus for our central chemoreceptors. So we will see how this carbon dioxide will cause an increase in the H plus concentration inside the blood-brain barrier, and how it causes an increase in the ventilation. So whenever the blood partial pressure of carbon dioxide increases, there occurs increase in the diffusion of the carbon dioxide from the cerebral blood vessels into the brain interstitial fluid as well as the CSF. And this carbon dioxide, it is promptly get hydrated to form the H2CO3 or the carbonic acid. And this H2CO3, it soon dissociates to form the H plus as well as HCO3 minus. The rise in these H plus concentration will stimulate the chemoreceptors. Thus, the carbon dioxide level in the blood will indirectly affect the H plus concentration inside the blood-brain barrier and produces an increase in the ventilation. Thus, the carbon dioxide level in the blood regulates ventilation chiefly by its effect on the pH of the cerebrospinal fluid. Thus, the carbon dioxide level in the blood regulates ventilation chiefly by its effect of the pH of the cerebrospinal fluid. So, the resulting hyperventilation, it will reduce the partial pressure of carbon dioxide in the blood as well as the cerebrospinal fluid. And the cerebral vasodilatation, which accompanies the increase in the arterial partial pressure of carbon dioxide, it will enhance the diffusion of the carbon dioxide into the brain interstitial fluid as well as into the CSF. When the blood carbon dioxide increases, the excitation of the respiratory center by the carbon dioxide is great in the first few hours. But then gradually this excitation declines over the next 1 to 2 days, decreasing to about 1/5 of the initial effect. Why? Why is it so? The answer is that it is partially because of the renal adjustment. That is, the kidneys will bring back the hydrogen ion concentration in the circulating blood back towards normal by increasing the blood bicarbonate, which will bind to the H plus ions in the blood as well as in the CSF to reduce their concentration. But even more important over a period of hours is that these bicarbonate ions, they will slowly diffuse through our blood-brain barrier and the blood cerebrospinal fluid barriers, and they will combine directly with the H plus ions adjacent to our respiratory neurons as well, thus reducing the H plus ions back towards the normal. So this is how there is a decline in the effect over the next one to 2 days as the blood carbon dioxide concentration is increased. Now we will see what is the effect of hypoxia on respiration. You all know that the normal arterial partial pressure of oxygen, it is 95 millimeters of mercury. A decrease in this PO2, it is a potent stimulus for which chemoreceptors? It is peripheral chemoreceptors, because hypoxia is the potent stimulus for peripheral chemoreceptors, whereas hypocapnia. It got a major effect on the central chemoreceptors. So whenever there occurs a decrease in the PO2, the peripheral chemoreceptors get stimulated. And if the PO2 falls from 100 to 60 millimeters of mercury, there is a significant increase in the afferent nerve discharge. But however, the pulmonary ventilation does not increase significantly because of the two reasons. We will see what are the two reasons which does not cause significant increase in the pulmonary ventilation when the PO2 falls below 100 to 60 millimeters of mercury. First reason is that this increased ventilation stimulated by the hypoxia will wash out all our carbon dioxide. So what happens when the carbon dioxide is washed out? The partial pressure of carbon dioxide will be decreased, and this will remove the stimulation of the respiration through its effect on the central chemoreceptors. And another reason is that when the arterial partial pressure of oxygen falls, it will cause less saturation of the hemoglobin. And so the amount of deoxyhemoglobin is increased, which is a weaker acid as well as a strong buffer as compared to that of the oxyhemoglobin. That is, it will bind with the H plus to form the HHB. This result in the mild decrease in the H plus concentration of the blood, which tends to nullify the hypoxic drive on the ventilation. Now, what happens when the PO2 levels falls below less than 60 millimeters of mercury? The stimulation of the peripheral chemoreceptors is so strong that it will override the inhibitory effect of decreased arterial partial pressure of carbon dioxide and decreased H plus concentration, and it will produce a marked increase in the pulmonary ventilation. Next is about the effect of hypocapnia on ventilation. So you all know that the normal partial pressure of carbon dioxide in the human body, it is 40 millimeters of mercury, and it is kept constant by our chemical regulation of respiration. And any increase in this arterial PCO2, it causes an increase in the pulmonary ventilation by stimulation of which receptors? It is by the central chemoreceptors, and to a lesser extent through our peripheral chemoreceptors. And there exists also a linear relation between increase in the partial pressure of carbon dioxide as well as increase in the ventilation. And stimulation of the ventilation will increase the washout of the carbon dioxide until the arterial PCO2 becomes normal to shut off the stimulus. And this ventilatory response to these changes in the PCO2 can be reduced by drugs like morphine, barbiturates, as well as the anesthetics, etc. Lastly, one word about the carbon dioxide narcosis. That is, when the carbon dioxide content of your inspired gas is more than 7%, what happens? The alveolar as well as arterial PCO2 will also rise abruptly in spite of the hyperventilation. And this accumulation of large amount of carbon dioxide in the body, that is hypocapnia, it will depress the central nervous system, including our respiratory center, and it can produce headache, confusion, as well as eventually leads to the coma of that patient. So that is about the chemical regulation of respiration. Thank you.